Hi, I'm George Hammond, conference producer for Technology Networks, and I'm here to introduce our last talk of the day, ultra-rapid development of safe and effective pandemic vaccines. I'm pleased to welcome our final speaker, Nikolai Petrovsky. Nikolai is Research Director of Vaxine, an Adelaide-based biotechnology company focused on vaccine development, and he is also a professor of medicine at Flinders University. He is a clinical endocrinologist and has a PhD in immunology. His work in vaccines and artificial intelligence has been awarded over $50 million in grants from the US National Institute of Health. He has authored over 200 peer-reviewed research papers and is an investor on multiple vaccine patents. A warm welcome to you, Nikolai. Following the presentation, we will have a Q&A session. We welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss a talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during this presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. And now, without further ado, I will hand over to Nikolai to begin his presentation.
Well, pleased to meet you all, and it's a great pleasure to be able to tell you some of our experience with developing a vaccine against COVID-19, which goes by the trade name Spikogen, and is now approved for use not just in adults, but also in children 5 years and older in the Middle East. So when we're developing a pandemic vaccine, speed is everything. Normally, a vaccine might take 15 years to develop. In the context of a pandemic, we're trying to compress that into a shorter time frame as possible, and ideally something more like 3 or 6 months rather than years. And in our experience, in that situation where speed is everything, nothing beats past experience. This is not something that you can do without lots of training, and in the vaccine field, that usually means not without having done a lot of research and development of different vaccines in the past. And we were fortunate back in 2002, in response to the original SARS outbreak, to actually apply for funding from the US government to develop a vaccine, and were successful in that. And as a consequence, back then we set about developing what we thought was a suitable platform for a vaccine against SARS, which was based on making a recombinant protein, which we expressed in insect cells and then formulated with our proprietary polysaccharide adjuvant, ADVAX, which I'll talk a little bit about later. And this proved to be a highly safe and effective vaccine in animal models. They were all completely protected against a challenge with a lethal SARS virus. So this was our starting point on a journey of almost 20 years now, developing vaccines against coronaviruses, as well as others that I'll talk about. So the next big coronavirus challenge was the Middle East Respiratory Syndrome virus, or MERS. This first emerged out of nowhere in Saudi Arabia in 2012, with a number of deaths in people. It was eventually traced to a virus that was infecting the camels, and prior to entry into the camels, there's evidence it came from bats, but it was essentially people coming into close contact with camels that were getting infected with the MERS virus, and with a very high mortality rate. About 1 in every 3 people who actually got infected by the virus, unfortunately, succumbed and died from the infection. So highly pathogenic virus. We were involved with the team in the US in developing a vaccine against MERS, again using the same approach of a recombinant protein-based vaccine combined with our proprietary adjuvants. And with the help of Professor Dick Bowen at Colorado State University, were actually able to show in real life camels and alpacas that we could use this vaccine to actually protect them against a challenge with the MERS virus. And you can imagine the logistics of trying to do give MERS to very large camels in very highly controlled biosecurity environments. It was, as I say, an extraordinary achievement, and that's now been published and is available. And then, so in early 2020, in fact, right at the beginning of 2020, I became aware through social media that there was an outbreak happening in China. And in mid-January of 2020, a sequence was released on the web of what was thought to be the virus causing these infections. And given my background in coronaviruses and pandemic vaccine development, of course, you know, I immediately set my team to work to try and study this virus from the sequence, because that's all we had at that point, and start designing a vaccine if we could. And at the same time, we also, because we were modeling this genomic sequence using supercomputers, which is one of our great strengths of our group, and using artificial intelligence to actually turn the sequence into the structure of the virus, we also were able to explore to a degree where the virus might have come from. And obviously, the hypothesis is that this came from bats through some intermediary animal host to humans. And so we were able to apply that technology to study what animals might have been involved. And that whole story, which to this day remains an intriguing story, we've published quite extensively on, is available if people are interested. But not surprisingly, we applied the same approach to developing a vaccine against COVID-19 as we'd previously done to SARS and MERS, hoping for the same outcome because we'd seen such strong protection with those earlier viruses. Just again to emphasize that the experience is what counts when it comes to developing a pandemic vaccine rapidly. This is just, it's not actually an exhaustive list, but a list of all of the different pathogens to which we have, with collaborators, developed vaccines over the last 20 years. And you can see there's very few, lethal pathogens out there that we haven't actually worked on and developed vaccines against. And this is because we don't know for the next pandemic at any point what virus or pathogen is going to cause it. And so the only way to be prepared is to actually develop vaccine platforms that are capable of tackling any and all of these pathogens, in the hope that when a pandemic does occur, we already have something in the cupboard that can be rolled out and adapted to create a suitable vaccine. So really, the experience that we've had, as a very small team, is that sometimes, being small can be a strength. It doesn't necessarily have to be a weakness. Most people expect that, in the face of a pandemic, only a massive vaccine enterprise, that will be able, that employs hundreds of thousands of people, will be able to move fast enough and be able to do all the work that's involved to develop an approved vaccine. Again, we are the proof that that's not actually necessarily true. And one of the strengths of a small, well-networked team is it's able to move extraordinarily fast when the need presents. I mean, from my seeing, the genome sequence made available in January 2020 on the web, to our scientists, starting to work on that was less than 24 hours. In a big company, the CEO might see something like that, but then he has to talk to the research director, and it may be 3 or 4 months before even the decision to start the project will filter all the way down to the scientists who are actually going to do the work. As I say, all of that is not true in a small group. And also to show you that I'm not just talking theoretically about the benefits of size, but if you look at our achievements as a small team and what we've been able to do in the past, I mean, in 2009, we had the swine flu influenza pandemic, and we actually had the first vaccine in the world, which was a recombinant protein, again, very similar to our current COVID vaccine platform, which we got into human clinical trials in Australia in under 3 months from first identification of that virus. At the time, that set a world record for development time of a vaccine from discovery of a virus to first human dose given. And, that record was only beaten, I think, recently by the mRNA vaccines in this pandemic that might have shaved a few days off it. But previously, I think the record was about 4 years from identification of a pathogen to first dose in humans. So we certainly took years off that previous record. Because we use a lot of artificial intelligence and always have in the last 20 years, when we push you small, you try to develop technologies that are able to allow you to do more than your normal size would allow. So we've always embraced artificial intelligence. And in fact, we believe that we have the first human drug, or in this case, a vaccine adjuvant, that was designed actually by artificial intelligence to enter human clinical trials. And this was part of a seasonal influenza vaccine trial in the United States, which started in 2019. Similarly, our COVID vaccine, we used artificial intelligence extensively in its initial design. And although it wasn't the first COVID vaccine in the world to enter human trials, in fact, I think it was either number 15 or 16, it actually was the first COVID vaccine that had been developed in the southern hemisphere to enter human trials, which it did at the end of June in 2020. It also emerged at the other end after its pivotal phase 3 clinical trial as the first recombinant full-length spike protein vaccine in the world to receive regulatory approval, which occurred in Iran in October 2021, which was a number of weeks ahead of the first approval of the Novavax, which is also a recombinant spike protein vaccine. So, as I say, just being small doesn't mean that you can't be first. When we look at the COVID vaccine landscape, I think that this has been, people's understanding of the landscape has been heavily distorted by media coverage over the last 3 years, which has largely focused on two of the early candidates that were given an enormous amount of airtime, these being the AstraZeneca adenoviral vector vaccine being driven out of Europe, and the Pfizer and Moderna mRNA vaccines driven out of the United States. And these really dominated, the Western media, to the point where I think people thought these were the only types of vaccines out there, and certainly, were led to believe that these were the only effective vaccines out there. But in fact, the landscape, the true landscape, was much more complex and had a lot more different elements to it. obviously there were the inactivated whole virus vaccines that were predominantly came out of both China and India, and with a recent European entrant from Valneva, which obviously were given to very large numbers of people around the world, particularly in the East. There was, the DNA vaccine, so one from Inovio in the US. There's also now one from India, which has in fact been approved. And then there's this category that really has been completely, overlooked, I think, certainly in the media, which is the recombinant protein-based vaccines, which is interesting because recombinant protein vaccines are the dominant vaccines that we've used over the last 40 years and continue to use, that are highly safe, highly effective. They form the basis of most of our pediatric vaccines that we give to our babies to protect them, but interestingly have been completely ignored and neglected in certainly the media attention on COVID vaccines, and I guess part of this is that whether it's our vaccine or Novavax's vaccine, neither of us are large companies. Novavax is somewhat larger than we are, but not in the scale of Pfizer, Moderna, or AstraZeneca, and so it does appear that science has not been what's shaped people's impressions or the media's impressions. It's been more the size and marketing dollars of the big companies promoting their particular technology. So what I'm going to focus on today really is to try and correct this imbalance and tell you why I think that protein subunit vaccines still represent the ideal solution, not just for COVID-19, but potentially for other future pandemics, and that we shouldn't be neglecting this class of vaccine the way that we have and dismissing it as a valid alternative technology to protect people against these types of pathogens. So just to drill down into a little bit of the basic science underlying our vaccine, which goes by the name of Spikogen or COVAX-19. So this was developed directly from the genomic sequence of the original Wuhan strain of the virus that was made available by Chinese scientists in January 2020. We had to make changes. So this is even before the virus had been described. We're already recognizing, for instance, this very unusual furin cleavage site that shouldn't have been in the virus because the related viruses don't have this, including SARS. So this mysterious furin cleavage site appeared and was clearly going to cause instability problems in the protein. So we set about actually removing the furin cleavage site and reverting the sequence back to the SARS sequence at this particular site, because we didn't believe this furin cleavage site was natural to these types of coronaviruses and could potentially cause problems, which in fact it did if you left it in. We also made a number of other mutations to the sequence that were planned to stabilize it. We had to modify the leader sequence because we're now going to express this protein in insect cells, which is not the native host. Obviously, it's normally expressed in mammalian cells when the virus infects humans. And also we had to codon optimize the nucleic acids, again, because the virus normally infects humans, and so it's optimized its codons to maximize expression in the human host, whereas here we're going to be expressing it in an insect cell. We also wanted to express it as a soluble protein. Again, this is based on our work with the SARS virus. We wanted it to be secreted from the insect cells, which makes it easier to purify and to make it a high purity product. So we set about removing the transmembrane domain. that anchors the spike protein in the cell membrane in order to make it soluble, but at the same time we wanted it still to form into its native trimeric structure, which meant that we had to be very careful that in removing this we still didn't, or that we didn't stop the protein being able to multimerize normally, and we're able to do that and simulate that in our models, show that this was working, and then actually express the protein as shown here. It runs as a single protein at about 180 kilodaltons because we've removed this furin cleavage site, so it's not cut. And if you look at the particles under negative stain electron microscopy, you can see that they form nice cone-like structures that correspond with the spike trimer, which is the proper native structure that we're looking for in the vaccine. This was then formulated with our proprietary polysaccharide Advax CPG adjuvant, which forms these small micron-sized particles that look like donuts that are very potent in inducing immune responses against co-administered protein. And then this is the look of the final vaccine, which looks like a milky white solution thanks to these polysaccharide particles. So the power of this technology is that, as I mentioned, we can go straight from the genomic sequence of a virus. We never actually ever have had COVID in the lab. I think the only time it's been in the lab is probably if one of our staff had a COVID infection. But you know, we don't ever have to handle the virus in order to make the vaccine, and this is great because if it was Ebola or something highly lethal, obviously we wouldn't want to be handling it. But what we're able to do is to take the genomic sequence as soon as it's available on the virus. We can then build a model of the virus. We can simulate it. We can identify what are the key proteins of the virus that are attaching to the host that's allowing it to infect. And then we can take that attachment protein, we can modify it, optimize it for expression, as I previously mentioned, and then simulate it using our artificial intelligence to make sure it's all working. If it is working, we then actually order a synthetic gene that corresponds to this. So this is just essentially printed. Again, doesn't require the virus itself. We take that synthetic gene and we now actually insert it into a virus called a baculovirus that can only infect insect cells. So this virus is harmless to humans or any mammalian hosts. It can't replicate at the temperature of mammals. Insects' temperatures are much lower, and that's one of the reasons it's so safe. We use this virus to then infect insect cells. It takes 3 days, and the insect cells over that time secrete the recombinant spike protein into the media. We then take the media, we purify the protein out of the media, and then it's formulated with our adjuvant, and here is the final approved product out of this process, which is the Spikogen recombinant COVID-19 vaccine, along with our adjuvant, which is mixed with it to make it highly immunogenic. So the benefits of expressing recombinant proteins in insect cells are that it's a highly adaptable system. It has a low cost of goods, much lower cost to culture insect cells than it is to culture mammalian cells. It's very scalable. So with our partner Synergen, this has been scaled to 3,500 litre fermenters, which are at the top end generally in biotechnology. And this means you can make millions of doses per batch. And as I said, the batches themselves are very quick to make in under 3 days from infecting the cells to harvesting the protein. The recombinant protein is stable under normal refrigeration at 2 to 8 degrees Celsius. So none of this, high or very low temperature freezers that are needed for mRNA. We can make very rapid strain changes because we can essentially change the virus and then infect the cells, and 3 days later we we have a new vaccine candidate. The technology has a strong safety record. Flublok, which is a seasonal influenza vaccine made in insect cells, is approved by the FDA in the United States for seasonal influenza, and so we know that this is a very robust and safe technology. And as I say, it's a technology that's cutting edge, but it's been around long enough that we really understand it very well. So in terms of cutting to the chase, or how well does a protein-based vaccine work? Does it actually protect against infection? Does it protect against serious disease? So this is showing you data which you can go to. It's available through the internet in this paper in Clinical Microbiology Infection that we published. And this shows that if you look at the survival curves of people in the pivotal phase 3 study, looking at infection rates in people who are on placebo and people who'd received active vaccine, you can clearly see these two curves progressively separating, indicating a much higher rate of infection in the people who hadn't received the active vaccine versus the active vaccine. So this at the time was all Delta, which was is to date the most virulent and most lethal of all of the strains of COVID-19 that we've seen. So obviously we were very pleased to have that outcome. We were interested to see, because we weren't dealing with the Wuhan strain, which was a much weaker strain that was around when all the other phase 3s of the mRNA vaccines were being done. We were interested to actually try and work out through various algorithms that have been published, how effective would our vaccine have been against Wuhan retrospectively. And so we ran, and again, this is in the paper if anyone wants to review the data, but we ran these algorithms that have been published, putting our antibody data against these algorithms to look at where we fitted on this curve which basically looks at antibody levels versus vaccine efficacy. And when we did this for Spikogen, using the various algorithms, you can see the algorithms all predicted very high levels of protection of Spikogen against the original Wuhan strain, ranging from 99% on this algorithm down to most of them sitting around the 90 to 95% level, which is about as good as any of the other vaccines published data. With just one algorithm suggesting somewhat lower, but still very positive protection at 64%. So again, suggesting that this vaccine is really, as a protein-based vaccine, is really up there in the same league as the mRNA vaccines, and potentially superior to the AstraZeneca adenoviral vector vaccine. And again, the animal data seems to put it in a very similar place when we do comparative studies in animals. Obviously, COVID has not stood still and continues to throw up new variants. And so the question then becomes not how effective was a vaccine 3 years ago against the Wuhan strain, which is obviously what the mRNA vaccines and adenoviral vector vaccines have largely keep quoting is that original data showing 90% protection for mRNA and 60 to 70% protection for adenoviral vectors against Wuhan. The real question now should be, well, what's their protection against strains like Omicron or Delta? Obviously, they're going to be much lower than against Wuhan. That goes without saying. So one way to look at this, and so this is our antibody data from our phase 2 clinical trial subjects, is just to look at the ability of that sera to cross-neutralize all of the more recent and more relevant strains. And so with Spikogen, what you can see is that in fact, we have high levels of neutralizing antibody against the original Wuhan strain, and these are maintained against all of the early strains like alpha and beta. Drops a little bit against gamma and delta and lambda, and then has another drop to Omicron. But nevertheless, we're still getting a significant number of people who have reasonable titers of antibody against Omicron, indicating a high level of cross protection after 2 doses. So as I say, this is just 2-dose data. When we look at the 3-dose data, we get a dramatic increase in the Omicron responses. So after 3 doses, they're not too different to the responses against the ancestral strain. So in terms of the current status of Spikogen vaccine today, so as I mentioned, in 2021, we completed a pivotal phase 3 clinical trial in just under 17,000 participants. I think the key outcome of this study is it showed over 77.5% efficacy against severe disease caused by Delta infection, and this was highly statistically significant. Remembering that a lot of the early phase 3 studies actually didn't show clinically significant outcomes in terms of reduction in severe disease, because their event rates, given they were dealing with the early weaker Wuhan strain, were too low to, even though there was a difference numerically, it wasn't significant. Whereas we were able, because we had a lot more events and they were being caused by a much more serious variant being Delta, we were actually able to show a highly significant reduction in events of severe Delta in the vaccinated subjects. We subsequently went on in October 2021 to get our first marketing authorization from the Iranian FDA for use of Spikogen as a primary course. This was later extended a few months later after we did a third dose booster study to include a booster dose. And just recently, we've received an additional approval after having done a pediatric study for use in children 5 years and above. To date, 8 million doses of Spikogen have been delivered. And despite that high number of doses, we have not identified any safety issues. In particular, we've not seen any evidence of myocarditis in people who've received Spikogen vaccine, which has obviously been a significant issue, not just with the mRNA vaccines, but also has been seen with the adenoviral vector vaccines, and also, interestingly, with the Novavax recombinant protein-based vaccine, but it hasn't been seen with Spikogen, fortunately. Obviously, we're now focused on development of multivalent vaccines. At some point, the virus will likely be able to escape the existing vaccines, and we want to be ready for that event. Although currently the original vaccine is still showing good protection, particularly against severe disease, including against Omicron BA.4/5, which we demonstrated in a recent monkey challenge study that was done independently by a collaborator in the United States. And we're currently awaiting WHO pre-qualification, pre-submission meeting, so we can look to start extending our approvals to additional countries around the world. So in summary then, this shows that 15 years of work can indeed be compressed into 12 months by a small team. I think we probably have the most extensive range of animal data showing efficacy of any of the vaccines. We've done this in mice, hamsters, ferrets, monkeys, all of the major models, and we consistently see protection, not just against the original Wuhan strains, but against Beta, Delta, and most recently Omicron BA.4/5. The vaccine's been through all the normal phases of human testing, and also post-approval studies have been done, including a booster study, a pediatric study, We have 3 different studies currently running in Australia looking at different dose intervals, and also a study in patients with renal transplants has been successfully executed in Iran. Just coming back to comparisons, because a lot of people have a belief that somehow mRNA vaccines are better than anything else, despite no comparative data really to show that. So it is really a marketing message rather than a science-based message, I believe. This is just looking at putting the vaccines head-to-head in mice. So here you can see data for mRNA vaccines and adenoviral vector and spikogen recombinant protein. These have all been given at 1/10 the human dose to make it a fair comparison. And what you can see is that the spikogen vaccines performing very similarly to the mRNA vaccines in terms of the level of antibodies against the spike protein receptor binding domain, whereas you can see AstraZeneca adenoviral vector vaccine very weak in terms in relative terms compared to the other technology. So this is reassuring us that we really are up there in the realm of the mRNA vaccines in terms of efficacy. Just to point out that, you know, there were enormous challenges. This was not an easy ride to get a vaccine developed and approved, you know, in around 18 months. This took enormous effort to get over all of the challenges. And in particular, because this was a new virus, you know, we had to develop our own reference standards, our own assays, our own animal models, our own manufacturing process, and we had to do this in a context where there was enormous disruption to the supply chain, which meant that some of our normal ingredients for our process, like processing bags, simply weren't going to be available for years, and we had to find instant solutions and workarounds for this. So it was an enormous challenge. And the challenge never stops. This is just looking at the variants of Omicron. And you can see that, you know, there's tens, if not hundreds, of emerging strains coming out of Omicron, and any of these could potentially escape a vaccine, and we might need to reformulate very quickly if a virulent new Omicron strain emerges. And of course, this may not and is unlikely to be the last pandemic we're going to face. There have been outbreaks of avian influenza just in the last few weeks, and so we need to be ready to pounce and start developing, you know, new vaccines if any of these threats do start to emerge at any point in the future. So we need to be prepared. We need to think about how to be prepared and what's needed, and essentially this is all about preparation, developing good platforms, which we believe we have with our recombinant protein-based insect cell platform. We need to build redundancy into the system, and ultimately, if we want to be fast, we need to integrate artificial intelligence into all the steps of the process, and so, you know, we can't be too prepared. I'm just going to quickly not really go into the artificial intelligence, but suffice to say, we've shown ourselves that you can apply artificial intelligence at just about every step in the process, from designing the vaccine to characterizing the virus to predicting what the virus will do in the future. So, you know, I think the future for AI in vaccine design is very bright. This is, again, just you can find in published data where we've published on this work, where we've applied artificial intelligence to all of these, to optimize all of these steps in the production process. And we've also used AI to actually identify new antiviral drugs on the sideline, showing the power of this technology to be applied all through drug and vaccine development. I'm just going to finish by showing you what can be done in terms of vaccine development. So this is our phase 3 clinical trial site in Iran. You can see that as we go through this video, that it was a highly custom designed site. We took over a luxury hotel and turned it into a clinical trial site with custom designed facilities, all electronic, cloud-based data capture with barcodes. And we were able to, with our partner Synergen, process 800 subjects a day through this single clinical trial site, which I think probably is a world record in terms of a custom site and ability to efficiently process patients. So just to finish, to say this couldn't have happened without our tremendous partnership with Synergen, which is one of the, if not the leading Middle Eastern biotechnology company. They produce a lot of monoclonal antibodies. This was their first time actually being involved in development of a vaccine, but really tremendous partners, and the success of this project really is owed to them. We have to acknowledge the tremendous support over the last 20 years of the US National Institutes of Health, who helped us to build this platform for pandemics. They were really true believers that this was a great platform, and so it was wonderful to prove them right by developing a successful COVID vaccine based on it. And then I don't have time, but obviously we need to acknowledge all our academic collaborators, our manufacturing partners like Pharma, and then a range of other entities that came in and supported us in many different ways to achieve this work. So thank you. Thanks to, as I say, all our partners, and I'm happy to take any questions.
Thank you so much, Nikolai. What an excellent way to round out the symposium. I can see that we have received some questions already, so I'm going to move straight onto the Q&A now. So your first question is, what are the relative benefits of proteins over nucleic acid vaccine technologies?
Look, that's a great question, and it's a question I think that is going to take time, and more time than today to answer, because we really don't know much about nucleic acid. I mean, this is a new technology. We're still learning about both its pros but also, importantly, its cons, and particularly some of the major side effects like myocarditis, which we're still trying to grapple with and understand their significance and even their cause. The great thing about recombinant protein vaccines is we have 40 or 50 years of experience in basically every person on the planet has had multiple protein-based vaccines. I mean, they are extraordinarily safe. They're extraordinarily effective. I mean, they've eradicated or close to eradicated a lot of diseases such as hepatitis B. So I think the real strength of proteins, even if the efficacy was similar, that the strength is that experience, the safety, that they're robust, they're reliable, and they ultimately have a low cost of goods. I mean, we can make our vaccine in the order of dollars per dose, certainly less than $10 a dose. And we keep hearing that mRNA vaccines, the price is being subsidized. But it's going to go up into the hundreds of dollars a dose in the very near future. So even on cost alone, I think proteins are going to win out. So, but in terms of the long term, which is better, really we have to watch that play out and look at where do nucleic acids fit. We still don't know their long term efficacy. We don't know their long term safety and what the significance of these side effects are, whereas we don't have those issues with protein. So, given a choice, personally, I might be biased. I would always go for the protein-based vaccine first.
That's a great answer. Thank you so much. In the interest of time, I'm going to carry on. So your next question is, what is the future of COVID vaccines given the rise of new variants?
Another great question. So again, we don't know, but we have to be prepared. What we know is that with each new variant, the level of cross-neutralization keeps going down, as I showed you on that early slide. So even within Omicron, we've gone from BA1 to BA2 now we're at XBB, 1.16 and all those variants I showed. Each of those new variants, the neutralization is lower. Interestingly, what we're seeing with our vaccine, I can't speak for the others, is the level of protection is not changing. And that's surprising because our vaccine is still based on the original Wuhan virus, from early 2020. So the protection against severe disease is maintained, which probably says that antibodies actually aren't mediating that protection. And so maybe we've been too focused on antibody, and again, that was driven by the mRNA companies. They do induce high levels of antibodies initially, potentially higher than with other technologies. And so not surprisingly, they wanted to focus everyone on those high antibodies. But I think the evidence is increasingly that those antibodies are not actually what's mediating protection. But at some point, it's possible that the virus will evolve, and our original vaccine may lose its efficacy, at which point we're going to have to modify it. We're already clearly working hard to make sure we have an alternative ready if that happens.
That's fantastic. Thank you. We've still got time for a few more questions. So your next question, Nikolai, is, do you think vaccines will need to change on an annual basis going forward?
So I refer back to the previous answer. I think, the answer is we still don't know. There is so much we don't know. I mean, I think, unfortunately, health authorities want to give people confidence, so they pretend we know lots of things we don't. But as a scientist, I know what I know, and I also know that there's a hell of a lot more we don't know. And so the answer is, that we don't know at the moment whether people need to have boosters or not. Certainly younger people who are healthy, there's a strong argument at the moment, at least, that boosters have not been shown to have really any benefit in that group. In the high-risk group, the elderly with multiple comorbidities, I think we'll just keep giving boosters just in the hope that they're going to reduce those people's risk. Again, the evidence of that is not terribly strong, but I think we're compelled to try and do our best. And so if we are going to give regular boosters, I think it will only be in those very high-risk groups. At the moment, there's really minimal evidence of benefit of boosters currently in young, healthy people. But again, that can change because the virus, we have to remember, is not standing still. It keeps changing. And so what I say today may be completely different to what I say tomorrow. And again, we shouldn't be pretending that we understand where this virus is going, because we don't. It's inherently unpredictable. Maybe artificial intelligence, if we can program it right, might be able to tell us the future of the virus, but I don't believe any human can currently foretell where this virus is going to go in the future.
That is a great answer. It's truly an ever-changing field. And we have time for one final question for you, Nikolai, which is, we seem to have evolved from being told that vaccines protect against infection and transmission. But now, sadly, that doesn't seem to be the case. So what is the ongoing role of vaccines going forward?
Again, really a wonderful question to finish, because I think a lot of people are disillusioned because, they really were told back in early 2021 when these vaccines were being rolled out, everyone needs to get vaccinated because that's the end of the pandemic. There will be no more issues going forward. It's just a matter of getting these vaccines in enough arms because they will stop infection, they'll stop transmission, end of story. And that hasn't transpired. I mean, even countries which are close to 100% of the whole population vaccinated, they still have the same size outbreaks as countries, in the developing world that, have used very little vaccine. So there's no evidence at all. In fact, there's negative evidence that the vaccines stop infection or transmission, particularly of the more recent variants. So where the vaccines, do have a role, and this should have been emphasized and communicated from the start rather than making this claim, which was not based on any real data, that they were going to stop transmission of the pandemic. The focus should have been on saying what they could do, which is to stop severe disease, which is a completely different use of the vaccine, because obviously if you're not at risk of severe disease, you could argue, well, I don't need to get vaccinated because I'm not at risk of severe disease anyway. And if that had happened, obviously we would have focused the vaccines on the high-risk individuals because they're the people at risk of severe disease. So I think going forward, media and health authorities need to come clean, admit that that was never true about the infection and transmission. It was more wishful thinking. It certainly wasn't based on any hard scientific data. And that was a miscommunication. Accept that, tell people that, get people to forgive them for that, giving that message that wasn't ultimately true, and refocus them on, well, but we still need the vaccines because people at high risk still need protection against severe disease. And that's where we're going to be focusing all our efforts going forward. But unfortunately, a lot of governments around the world have been slow to realise that's what they have to do. They have to come clean in order to reposition the vaccines so that the people who need to get them don't stop getting them. As I say, there is this backlash that they're going to have to deal with. The best way is to come clean, admit your mistake, and refocus people on what the true science shows, which is they do have very great benefits in preventing severe disease.
That is perfect. Thank you so much, Nikolai. Unfortunately, that is all we have time for today, but it's been an absolute pleasure to have you present with us. Any questions that we didn't have time to get to will be answered offline as soon as possible. So that's it for Cell and Gene Therapy 2023. As mentioned throughout the day, all the talks from today's event will now be made available to view on demand for the next 30 days. You can access the talks via the same link you used to access them today, and we will be sure to pass on any further questions you submit through the system to the relevant speaker. If you've enjoyed all of the excellent talks over the last 2 days and want to hear more exciting cell and gene therapy content, then why not register for our upcoming Innovations in Cell and Gene Therapy online event. On June 14th, Miltenyi biotech researchers and customers will be coming together to discuss the cutting-edge technologies in this field and how it helps overcome challenges in cell and gene therapy development. If you would like to register for your free place, please click the link below this live stream panel. And that just leaves me to say thank you once again, and we'll see you again soon. Goodbye.